Colorectal cancer is one of the most prevalent cancers in humans, and about 10 to 20% of stage 1 to 3 colorectal cancers are classified as microsatellite instability high. Microsatellite instability is a genetic condition that occurs due to impaired DNA mismatch repair system in cancer cells, resulting in high mutation rates in microsatellite sequences. As a result, highly variable microsatellite mutation profiles can be observed among cancer cells. Retrospective lineage tracing is a type of lineage tracing that relies on naturally occurring genetic markers to infer cell lineage. Retrospective lineage tracing has been attempted by profiling microsatellite mutations on single-cell DNA sequencing of HCT116, a highly metastatic colorectal cancer cell line with microsatellite instability phenotype. However, lineage tracing based on DNA sequencing alone cannot reveal the transcriptional heterogeneity among the profiled cells, which is generally of great interest in cancer research. To address this limitation, we used nanopore long-read sequencing to analyze single-cell transcriptomes and reasoned that sufficient number of microsatellite regions could be profiled in each individual cells for high-resolution lineage tracing. This would provide new insights into the subclonal structures of microsatellite instability high colorectal cancer cells and their response to immunotherapy. Using CRATE, a robust tool for identifying microsatellite in a given genome, we first obtained a list of about 1.2 million microsatellite regions within the GRCH38 genome, among which about 500,000 regions were overlapped with REF-seq genes, as shown in Figure 1. Using 10x chromium single-cell 3' kits, we captured the transcriptomes of about 6,000 HCT116 cells. We sequenced the single-cell full-length cDNA library using an Oxford Nanopore Technologies Promethean device and generated 32 gigabase pairs of nanopore sequencing data. The same library was used to prepare a conventional short-read single-cell RNA sequencing library, which was sequenced on an Illumina HiSeq device. We developed a bioinformatic pipeline to analyze single-cell full-length RNA sequencing data and output a barcoded BAM file. We applied our pipeline to long-read sequencing data and observed a high correlation between long-read and short-read single-cell sequencing results as shown in Figure 2. Using single-cell long-read sequencing data of HCT116 cells, we discovered about 97,000 microsatellite regions were covered by transcriptomes of HCT116. We further filtered the microsatellite regions based on the number of molecules covering the site, the numbers of cells harboring the molecules covering the site, the number of detected alternative alleles, and the length of tandem repeat motif, and obtained 5,311 highly informative microsatellite loci as shown in Figure 1 and Figure 3. We further classified these informative microsatellite loci into 6 categories based on their insertion-deletion tendency and the effective number of alleles as shown in Figure 3a. We observed the majority of informative microsatellite loci can be classified as regions with deletions, which is in agreement with the previous observation that microsatellite regions tend to undergo deletions in microsatellite instable tumors. However, we also observed a significant number of microsatellite regions with strong insertion tendencies, or both insertion and deletion tendencies, as shown in Figure 3c and 3d. We hope our analysis pipeline can be utilized to select informative microsatellite loci to increase the cost-effectiveness of retrospective lineage tracing of single cancer cells using long-read RNA sequencing. Thank you for listening.